All right, so I've got lots of introductions, so I'll maybe skip it. So this is Jim's slide of photosynthesis. So electrons come out of water and via rubisco into organic molecules. And so far this meeting has been mainly on this bit. But biology, it has, for every carbon, there's quite a lot of nitrogen as well. And photosynthetic electrons are used directly or indirectly to drive biological nitrogen fixation. And photosystem 2 releases oxygen, and oxygen inhibits nitrogenase. So there's both necessary processes, both in conflict. And for biotech purposes, we would quite like to be able to put more electrons from photosynthesis into nitrogen, because a lot of photosynthetic light harvesting goes to waste. Maybe we could put those electrons into nitrogen. So that was the motivation. So electrons can go into nitrogen to make ammonia, and hence via glutamate into biomass. Okay. So this all came about because working on PS2 with Jim, and he asked me the question, how conserved are the OEC residues? So Gary started with exactly this similar question. So all of these residues make specific interactions with the manganese cluster, and they're all basically conserved between spinach and cyanobacteria, although surprisingly tolerant of mutation. And Jim asked me, James, do you think, can we just look and see if they really are conserved? And the answer is no. So Gary mentioned lysine 317. We've just been talking about that. So I looked at one of my old talks. There is one cyanobacteria where that lysine is an alanine, which is Gliorbacter violaceus. And when I looked at this a few years ago, this was the only variant I found in the database in 2,000 sequences. And so this Gliorbacter is this lysine is probably something to do with proton exit, and it does bind the chlorine at the site. So Gliorbacter is strange because it's not normal. So this is a normal cyanobacterium. It's got a thylakoid membrane with a pH gradient across it, and the PS2 the chemical protons must exit into this thylakoid lumen, which is possibly a difficult step. In Gliorbacter, there is no thylakoid membrane. There is no thylakoid membrane. The PS2, the protons exit straight into the periplasmic space. So maybe acidifying this, essentially the Pacific Ocean, is easier than acidifying the thylakoid lumen. So maybe there's some reason why that might be. But the main thing I looked at was D1. So as I said, these are surprisingly tolerant of mutation. So things like aspartate 170 to histidine or glutamate 189 to arginine, which are pretty non-conservative changes, still evolve oxygen, although you never ever see these in biological sequences. They're not as good, but you might think that there would be conditions under which different residues would be optimal. Particularly as if you look at cyanobacteria, they all have more than one D1 sequence. So almost all cyanobacteria have about 3 to 6 D1. So this is the protein that binds the OEC. Chloroplasts get away with generally only one. And multiple PSBA, which is D1, has evolved independently multiple times. So this is, for example, Thermosynechococcus elongatus. It's got 3 D1 genes. Seneca cystis has got 3 D1, and they branch within Seneca cystis and within elongatus. So it's not just gene dosage. They have, so this is a very highly expressed gene, but it's not just the dosage. They have different functions. So under high light, a particular one is expressed. Under microaerobic conditions, another one is expressed. So this is like different tools that fit into your screwdriver. You have a different head for a different function. So the upshot of the search for are there non-conserved OEC residues was yes, and they were all in a special class of D1, which I called rogue D1 because it looked as though it was doing the wrong thing. Other people in the literature have said they don't like that name, so the Picrazzi group call it Sentinel D1. And how they look, well, we don't know how they look, there is no structure, but this is the normal photosystem 2 with the OEC and the conserved residues around it. The rogue D1 looks like a pseudo-enzyme, so it has changes that look like they're not just different, but they completely abolish oxygen evolution. So for example, glutamate 333, which is a bidentate ligand to the manganese, is changed to alanine. And if you make that in Seneca cystis, that abolishes oxygen evolution. Aspartate 61 becomes a glutamate. Aspartate 170 becomes a glutamate. Glutamate 189 becomes an aspartate, which is also a change that knocks out oxygen evolution. Aspartate 342 becomes threonine. So we don't really know what metals bind here, but definitely not an OEC-style oxygen-evolving cluster. So that's at the OEC site. If you look at residues that are consistently variant in these sequences, so I've drawn those in purple, so this is in the sequence alignment, variants that are consistently different from a canonical D1 sequence. They are clustered around the OEC, as I've just shown, and also around the QB site. So there are absolutely conserved residues here that are changed. So this doesn't bind, looks as though it doesn't bind a manganese cluster, and it probably doesn't bind the exchangeable quinone QB either. So it's looking non-functional. They are phylogenetically ancient, so the normal variant D1 seem to be evolved within a particular species. If we root the tree on D2, which is homologous to D1, we see that these rogue sequences are older than all of the other extant oxygen-evolving D1s that we have sequences for. So they seem to be as ancient as any oxygen-evolving PS2. So whatever they're doing, they seem to be inactivating PS2, and they seem to be pretty important because they're old. The sequences are more variant. So this is a phylogenetic tree from Tanai Cardona, who's at Imperial. These are the normal, he classifies them into different groups. These are the normal D1, so they're short branches, and these are the rogue sequences. They have much longer branches, so there's more variation. So I would say that this probably means that functional constraints on the sequence have been relaxed. So it has more, whatever it's doing, it's got more freedom to vary. And if it's not evolving oxygen, maybe that's a lot of freedom. So when I was doing these sequence alignments, I thought, well, what have these organisms got in common? They look as though they evolve oxygen. What is it that is inhibited by oxygen? So looking down the list, in 2012 it was something like 19 out of 20 sequences were diazotrophs. Now that would be harder to spot, because if you look, there are many more cyanobacterial genomes, so picking out the phenotype is harder. But basically the thing that these all have in common is that they fix nitrogen. They are diazotrophic. So that kind of makes sense. And if we look at this transcriptomic study in light-dark cycles during the day, photosystem and carbon fixation genes are expressed, and at night, respiratory genes and nitrogen fixation genes. So this is the yin and the yang, the light and the dark. But strangely, in this same set, so these are there are 4 D1 genes. The 3 normal ones are expressed more in the light and less in the dark, more in the light. The rogue D1 is transcribed in antiphase, so there is more of it at night, which is strange for a photosystem gene, and less of it during the day. This is another transcriptomic study on Crocosphaera, which shows the same thing, that the PSBA4, which is the rogue sequence, is expressed in the dark cycles and not in the light cycles, whereas the normal D1 genes are expressed, as you would expect, in the light and not in the dark. So that's very nice. And we are currently trying to make these rogue D1 PS2s to study them and see if that's really true, and if they do any chemistry at all, or if they just turn off PS2. So that's temporal separation. Another thing that cyanobacteria do is they spatially separate nitrogen fixation from photosynthesis. So they make differentiated cells called heterocysts. But Tanai Cardona showed that some of these do in fact contain PS2 genes, and some of these have a rogue D1. So it might be that these, under some conditions, heterocysts will express a PS2 and then turn it off with this sequence. So this ROTE1 this is a summary of this section, is apparently as ancient as current oxygenic PS2. It has greater variation, which we assume reflects fewer restraints on functionality. So the hypothesis is that it maintains an inactive PS2 complex structure. So it's a structural D1, but not a functional D1, that suppresses oxygen evolution so that nitrogen fixation can proceed. Okay, so that's that section. After this, I spent a lot of time talking to Bill Rutherford about PS2 and nitrogen and nitrogenase and where the electrons go. So we have started looking at nitrogenase. So this is the energy of formation of water, so we have to reverse that. Fixing nitrogen is harder because you have to break this triple bond, which is nearly 1,000 kilojoules per mole. That's the second strongest diatomic bond known. The other is carbon monoxide, so it's a lot of ATP to hydrolyze to break those bonds. Industrially, we do this with the Haber-Bosch process. Thermodynamically, that's actually not too bad because hydrogen is a good reductant, and this is energetically fairly flat. But to break that bond, you need to put in a lot of energy, so it's done at 200 atmospheres at high temperature. And this uses several % of the global methane supply, so it's a big drain on our resources. I would say, although it is a big drain, it's actually a bargain because it has doubled the flux of reactive nitrogen into the biosphere. So all the carbon we burn is only changing the carbon flux into the atmosphere by a few %. We have doubled the flux of reactive nitrogen into the biosphere, which is causing lots of pollution and bad news. So it would be nice if we could increase biological nitrogen fixation, which is much less polluting. So the nitrogenase enzyme does similar chemistry. It's an 8-electron reduction to the Haber-Bosch process. It's an 8-electron reduction of nitrogen with 16 ATPs, at least 16 ATPs to ammonia, with an obligate production of hydrogen. So just on stoichiometry, that's twice as expensive as carbon fixation. The real cost is probably much higher because this ATP is a minimum, and this thing has to be kept anaerobic, which also increases the cost. And there's an obligate production of hydrogen, which has to be recovered. Like PS2, nitrogenase is complicated. It has only 3 structural genes, but it has a whole suite of assembly factors to make these metal cofactors, which do the reduction chemistry. So it's got 2 components, the molybdenum iron protein MoFe, which has the FeMo cluster where nitrogen is reduced, and the iron protein, or NIFH, which is the ATPase that accepts electrons from ferredoxin and delivers them to the MoFe protein. So the MoFe cofactor is maybe even more complex than the oxygen evolving center, the manganese cluster. So this is the structure. These are very reduced clusters, and they react strongly with oxygen. So nitrogenase is inactivated by oxygen. These reduced iron sulfur clusters react with oxygen. So the half-life in air of these proteins is short. The iron protein, only 30 seconds, and the MoFe protein, 10 minutes. So much of the biology of nitrogen fixation is involved in keeping these things apart. So if we're interested in improving nitrogen fixation, one thing to think about was, can we improve the oxygen tolerance or resistance of nitrogenase in some way? Maybe we could look at the ROTE1 in cyanobacteria. So nitrogenase is found only in prokaryotes. If a eukaryote is fixing nitrogen, it will be with a symbiote, which might be an endosymbiote. We would like to express nitrogenase in plants so we can reduce fertilizer usage, and they're aerobic eukaryotes. Cyanobacteria emit oxygen. How is that compatible with their nitrogenase? So in the first part, I gave working towards an answer, which was they can turn off their photosystems. the model Azotobacter vinelandii is an obligate aerobe. How does that defend against oxygen? Okay, so I'll take a little break on nitrogenases. Azotobacter has the 3 kinds of nitrogenase. So I showed the MoFe, which is molybdenum. There are 2 other enzymes, vanadium and iron only, which replace the molybdenum with vanadium or iron. These have less activity and are more sensitive, even more sensitive to oxygen. There was a report in the literature of a superoxide-dependent nitrogenase, which was completely different and homologous to a carbon monoxide dehydrogenase. So this was from Ortvin, Meyer, and Bayreuth, where he has a nitrogenase, which is, takes electrons from carbon monoxide to superoxide. So superoxide is the donor, and then nitrogen is reduced. So I spent a lot of time, too much time, working on this. It's kind of superficially plausible because, as I said, nitrogen and carbon monoxide are both they're isoelectronic and they're very strong trivalent bonds. The carbon monoxide dehydrogenase has molybdenum. It's bound to a molybdopterin cofactor, so it's like a flavin. So this thing has molybdenum, and it does chemistry to a strong diatomic bond. So it's tempting to think that maybe it's plausible that this enzyme, which is the aerobic carbon monoxide dehydrogenase, has nitrogenase activity. As I say, there's a lot wrong with this work. I'll just show you one piece that's wrong with it, which is this is a student thesis from the lab that did the work. This is a ion exchange chromatography run, and they have the protein coming off at the open circles, and they assay the fractions for carbon monoxide dehydrogenase activity, and they see a nice peak of activity. So that's the enzyme. They measured nitrogenase activity via ammonia production. So if you look at this is the open circles are again protein, and the closed circles are ammonia production. And I think they took these fractions as the nitrogenase, but actually the ammonia production is basically following the protein. So I would interpret that as a protein deamination is where the ammonia is coming from, and that's an artifact. Yeah, so that culminates in this paper. If you want an amusing read, read the discussion section of this. So we were very excited by the possibility of an oxygen-tolerant nitrogenase, and so we were, even though it was a little bit questionable, we pursued it really to the end to prove that this thing did not exist. So we are stuck for nitrogenases with oxygen-sensitive nitrogenases. So that one's dead. Okay, so how do diazotropes protect themselves from oxygen? So this is Azotobacter, the model 1. It covers itself in this slime, which I think is part of it. Apart from that, there are 2 main mechanisms. Respiratory protection. Azotobacter has a very high respiratory rate. So if it consumes oxygen quickly, that decreases the oxygen concentration. It also has this mechanism, conformational protection, where a protein forms an inactive complex with nitrogenase, which is inactive but resistant to oxygen. Okay, so I'm going to talk now about a protective protein that we investigated. So in the Azotobacter genome, there are lots of multiple copies of respiratory proteins. So there are 2 ATP synthases. There is a cytochrome BD terminal oxidase. I think there are other alternative respiratory proteins as well. So we investigated another one so far unpublished called ANF3, which is a novel flavor cytochrome with a probable role in oxygen protection of nitrogenase. So in the 90s, Bob Eadie's group identified a cytochrome that co-purified with the iron-only nitrogenase. They didn't clone it, they just purified the protein, and from that they got an N-terminal sequence, which was in one of his students' theses. Using that sequence, you can identify the gene in the genome, and this is it, cytochrome B prime. It is just next to the operon for the iron-only nitrogenase, so that's kind of a smoking gun that it's probably relevant because it co-purified with the iron-only nitrogenase. Because there are lots of genomes, we found this gene in other genomes as well, where it had been annotated as ANF3. So these are other iron-only nitrogenase operons with this ANF3 gene. There is a paper where if this is knocked out in Rhodobacter capsulatus, you abolish iron-only nitrogenase fixation. So this thing is important. So we investigated it, we cloned it, purified it, crystallized it. We got nice red crystals because it's a cytochrome. We got a nice 1 angstrom structure, and it's a dimer. To our surprise, we see the heme, and just next to it we see a flavin, which was a bonus cofactor that we weren't expecting. We maybe should have been expecting it because phylogenetically, if you put this protein into BLAST, it is a flavin-binding split-barrel protein, and there are lots of homologs in the PDB, none of which bind heme. It's a flavin protein that's learned to bind heme rather than the other way around. So we see that this is the heme there, and the flavin is pi stacking. Flavin FAD is pi stacking against it. We see a water bound to the heme, not an oxygen. With the eye of faith, you can just about believe that you can see the spectral change from the FAD upon reduction. We definitely see the heme. So this is probably to do with overlap from the heme and then interference because it's so close. It binds carbon monoxide. So we did a spectropotentiometric titration. So the heme has a potential of minus 340 millivolts and the FAD of minus 415, minus 420 or so. So these are rather low potentials. We see, because of the close interaction of the cofactors, we see a distortion from pure one-electron Nernstian behavior. So from all this, we thought this maybe could be could donate 4 electrons to oxygen. So skipping ahead, that's what we found, and I'll now show why we think that. So we came up with an assay where we used ferredoxin NADP plus reductase, FNR, to use NADPH to make reduced ferredoxin. And we tested that with ANF3, or cytochrome B prime, and also with cytochrome C as a control. So cytochrome C will accept electrons and become reduced, but go no further. ANF3 may or may not give electrons to oxygen and produce water. So under anaerobic conditions, in this assay, the cytochrome C is rapidly reduced, as one would expect. Our protein is more slowly reduced, but the heme does become reduced. Under aerobic conditions, the cytochrome C again is rapidly reduced and stays reduced because there's nowhere for the electrons to go. The heme stays oxidized, so presumably any electrons that go to the heme are immediately going to oxygen and thence to water, and that's where that's going. In the oxygen electrode, we see that in the assay conditions, when we add ANF3, the oxygen concentration is reduced. I will skip ahead quickly through this, but we look at superoxide production. With our protein, we see less superoxide produced. So the assay generates superoxide, but with our protein, we see less. So it's not generating superoxide. We also used Amplex Red to look at hydrogen peroxide, and we don't. This is this. So we see less production of hydrogen peroxide with ANF3 than in the control. So we think it is going directly to water, not to hydrogen peroxide or superoxide. So we think electrons are coming maybe to the FAD, then to the heme, thence to oxygen. Where there are 4 electrons required, where do they all come from? So 2 of them can come from the FAD and the heme. There are tyrosines nearby, which could donate electrons. There is a network of waters through which protons could come. Because it's a dimer, the FAD is actually only 15 angstroms away from the other one, so that's another source of electrons. Okay, so to summarize ANF3, electron transfer occurs from a donor to the FAD, then to the heme. And our structural and biochemical data shows that ANF3 reduces oxygen to water in a 4-electron reaction. Yes, this is a ferredoxin oxygen oxidase reductase. It's an unusual protein. It's a flavor cytochrome. The heme and the FAD interact very closely. We assume that the heme has a high affinity for oxygen, so this is a kind of high vacuum pump for oxygen. It maybe has the same low potential donor as the nitrogenase itself. From this biochemistry and the associated phenotype we see in the mutant, we would guess that it's protecting the iron-only nitrogenase from oxygen and maybe has some application in heterologous expression of nitrogenase. Okay, so I've reached the end. So the summary, I told you about the ROGD1, which is a photosystem 2 pseudoenzyme that we think turns off oxygen evolution. And I told you about nitrogenase and an oxygen reductase that we found that protects the iron-only nitrogenase from oxygen. Okay, thank you. And I'd like to thank my group, particularly Febin, who did all the ANF3 stuff with Andrea and Bill Rutherford. So, thank you.
Yeah.
I think it was Holger and then Johannes.
It's an extra burden on me. I can carry this burden much further. Does anybody want to take over as chairperson? Could I ask somebody to be a chairperson instead of me? Peter, could you?
Yeah.
Okay.
Yeah.
Thank you. I'm very dodgy at the moment. Okay. Okay, I'll just settle down. Okay, any questions?
Yeah, I already got the microphone somehow. So, I have a question about these ROGD1 forms. Okay. Do they all, there's a lot of variants I figured from the literature, from Imperial College literature, and so are they all inactive or predicted to be inactive also at the acceptor side?
I think they are. So every time I look, there's more sequences. But as far as I can tell, they all. So some of them even don't look as though they bind a non-heme ion either. So they have. So I think they're all predicted to be inactive at the acceptor side. So it doesn't look as though they have an alternative donor that's not water, if that's what you're getting at.
Okay, yeah, right. Mm-hmm. Okay, and are there any functional studies aside from this computer studies?
Yeah.
On sequences?
So. Pecrazi has a paper where they express them. They don't see any oxygen evolution activity. I think we are currently trying to express them because we would like to purify it and look at it more closely. There is, of course, the class that I didn't talk about, which is even stranger, which is the putative chlorophyll F synthase, which is to Don Bryant's work. Yeah.
Okay, right. Thank you.
Yeah, I'm also fascinated by these inactive D1 proteins. So why in the dark do they need also an inactive D1 protein? I mean, wouldn't you expect PS2 to be non-active anyway?
So I think that these lab experiments are light dark, and in real...
The moonshine.
So I think in real life it's more sinusoidal, and I think there are probably conditions under which it is light enough to activate PS2, but maybe dark enough that the organism doesn't want to. Having said that, the expression levels are not that high and probably not enough to make all of the PS2 this variant form. Yeah. So one of the reasons we would like to purify it is perhaps it is some sort of signal that if one is changed, it somehow inactivates others, because I think stoichiometrically there's not enough to inactivate the entire pool of PS2.
Okay. Interesting, anyhow.
Yeah.
Yeah.
Okay, there's a question.
So I'm wondering for this protective enzyme against dioxygen, at what level, what concentration relative to nitrogenase, it's leaves the nitrogenase competent for its function?
Yeah, so We don't know because our assay is not really physiological conditions, so we're not. Our donor is spinach ferredoxin, and the potential of the actual ferredoxin is lower. So I'm not sure about the rates. I think the affinity for oxygen is probably, I want to say micromolar, but we need to measure that. It's hard to measure because, yeah.
Okay, one last question.
Yeah, the question I was wondering, is the nitrogenase expression going up when your ROAT D1 expression goes up? Is that coincidence? Is that at the night happening only, or?
Yes, so that's, I haven't drawn the individual genes, but in the basic bar chart, all the nitrogen, the NIF genes were upregulated at night. So.
Do you have also Schnittner protein that protects additionally?
Yeah, so I actually have I didn't have time to talk about it, but I actually have a structure of the Schnittner protein. So I can talk to you about that if you want as well, but yeah.
Okay, can we thank James yet again for a very nice talk?